So I'm a scientist at DNR, and I lead our research program into ocean acidification and climate change across state-owned aquatic lands. So not specific to Cherry Point, but I'm going to try and relate some of our work to this particular site and explain some of the collaborations that we have going with the Aquatic Reserve system. So I'm going to start with an overview of something that probably a lot of you in this room are following closely, which is on ocean acidification. So who in the room is following ocean acidification as an issue? All right, this will probably be a basic overview then, but just so we're all on the same page. Ocean acidification is the other CO2 problem besides climate change. When we emit carbon dioxide into the atmosphere through human industrial activities, a fair amount of that carbon dioxide, which is diffuse in the atmosphere, dissolves across the atmospheric ocean boundary and changes the chemistry of overlying waters. So this is a pretty substantial process on the scale of global carbon cycling, because since the start of the Industrial Revolution, the oceans have soaked up about 30% of the carbon dioxide that we have dumped into the atmosphere. So failing that absorption, climate change would actually be more severe than it is now. And the oceans are going to continue absorbing the CO2 at the same rate. So we can predict into the future, as the oceans come into equilibrium with the atmosphere, what that'll mean for chemistry. And through just basic reactions of CO2 with water, the carbon system changes. So when CO2 in its gas form dissolves into the ocean, we have an increase in hydrogen ions, which is the definition of a decrease in pH. The ocean becomes more acidic, and an increase in that term on the right. This is pretty much all the chemistry that's in the talk, so just bear with me, is an increase in the bicarbonate ion and a decrease in the carbonate ion. So higher atmospheric concentrations of carbon dioxide increase the proton concentration, hydrogen, and decrease pH. And this is what people mean when they talk about ocean acidification. To date, from pre-human levels of carbon dioxide in the atmosphere, which were about 280 parts per million, we've just crossed the threshold of 400 year-round. The oceans have decreased on average by 0.1 pH unit, which is about a 30% increase in the hydrogen ion concentration. So where we know this from, the strongest documentation is from the Mauna Loa Observatory in Hawaii, and it has provided really strong evidence of both changing carbon dioxide concentrations in the atmosphere, which you see here in the red curve. This is the famous Keeling curve that was started 50 years ago. And then CO2 concentrations in the ocean, which is the curve right below that. So as you see, changing concentration of CO2 in the ocean as it goes up, this light blue curve shows pH going down. So this is what's happening in open ocean systems far away from Washington State. And the question is what it means for environments closer to shore and on the continental shelf in Washington. And there's a lot of science underway right now to connect these observations in the middle of the Pacific Ocean with what we're experiencing here. And for me, as a biologist, the primary question is what this means for biological function and for the performance of the plants and animals that are under DNR's management purview. So here, maybe some of you have heard of these animals, is a picture of a pteropod or a sea butterfly. They have kind of become the canary in the coal mine in ocean acidification science. They're beautiful little animals, and let's see if video translates. All right. So these animals are actually related to clams and snails, even though they don't look like it. They're planktonic, so they live in the open ocean, and they're called sea butterflies because they have those two little wings on top that they use to swim through the ocean. They have a small shell, which is probably vestigial in nature. It's just because that's their evolutionary origin. And the fact that they have that shell means that they are sensitive to ocean acidification and have provided some of the most robust science to date about what decreasing pH means for animals living in the marine habitat. So to show you in picture form what that means, experimental work has shown that here is a pteropod shell as an electron micrograph. Hopefully you can see this. And magnifying that area, you can see what the shell margin looks like for an animal that's raised under low pH conditions. And in comparison to the shell margin for an animal at pre-industrial levels of CO2. So you see really clear visual evidence of shell dissolution, that the animal has trouble building this protective shell in low pH conditions, and that probably means something for its function in nature. We're not sure exactly what it means, but it's become a point of focus in the news because the change visually is so dramatic. So to sum up what that means, pH is a master variable, and one of the first things that we expect it to change in marine organisms is the rates of calcification. And for an animal to perform calcification, it has to pull carbonate ions out of the water, combine them with calcium, and create calcium carbonate, or chalk, to build its shells. And that's what all bivalves do in one way or another, build a shell out of these two ions. And if you'll remember the chemistry slide, as the pH of the ocean is going down, there's less of this first reactant of carbonate ion. So it's both increasing the rate of shell dissolution by the pH being lower, and it's decreasing the bioavailability of the carbonate ion, which the animal needs to put together with its shell. So what other animals besides pteropods, which live around here, are going to be sensitive to ocean acidification? If we just look at the groups of animals that depend on calcification in particular, the ones that come up first are corals, stony corals, coccolithophores, which are kind of phytoplankton that have these beautiful small shells made of calcium carbonate. Brittle stars and other echinoderms have calcified structures over their bodies, so those include sea urchins, sea stars, brittle stars, and crinoids. And close to the economy of Washington State and the management interests of DNR, geoducks and oysters build shells that protect them from predators, and their rates of calcification may change with changing ocean chemistry. So I just wanted to talk about the pteropod story because it's in the news so frequently, and then to really briefly sum up the explosion of science that's happened on ocean acidification impacts in the past 8 or 10 years. It's not all a clear story. Pteropods are one where we see lower pH means dissolved shells and these kind of jagged shell margins. But other experiments on phytoplankton like these coccolithophores, which are here under an electron micrograph, show that it's going to be hard to predict at an ecosystem level what ocean acidification means. So in the laboratory, when these kind of impact studies have been done, and this to date has been the most common kind of ocean acidification science, you get an animal that you're interested in, you go into the laboratory, you have a tank with a particular concentration of CO2, you drop the animal into it and then pump up the CO2 and see what happens. It's pretty basic science. But coccolithophores, where you might expect there to be a big effect of pH and carbonate ion availability, show a sort of unexpected pattern. So here is a coccolith. It's the little piece of the shell that the phytoplankton builds under pre-industrial conditions of CO2, 280 parts per million CO2. Here it is at 300, 480, so that's above where we are now, 595, and 750. So this is one of those experiments that kind of is painting a complicated picture of ocean acidification and defying the ability of scientists to tell a unified story about biological effects. We see that with increased CO2 concentrations, coccolith deposition rate increases and final volume is higher. So these organisms benefit from a higher CO2 level in the ocean. And as we're delving into the physiology of calcification, the answer appears to lie in the fact that calcification pathways don't always use the exact same reaction, that different classes of organisms have the ability to concentrate carbonate ions to fight against the effects of ocean acidification, and some actually derive the carbon that goes into their shells not from seawater but from internal physiological processes, and that is probably the case with coccolithophores. So this is all just to say that some organisms that build shells are probably going to be more resistant to ocean acidification than others, and it's because there are a lot of diverse ways that animals build those shells just through basic chemistry. Yeah, absolutely. yeah, if I go back to a picture of a whole bunch of these guys lying around. So these are a bunch of coccolithophores, and their shell isn't one thing. It's pieced together from a bunch of different coccoliths. You can kind of see them all overlapping. And at higher CO2 levels, those individual coccoliths are bigger, and that leads to a larger overall shell and higher volume. It's a really good question of what that means ultimately, because this experiment did not look at anything like fitness or ultimate reproductive success of these organisms. So all we know is that acidified conditions resulted in larger organisms with more robust shells. We don't really know if that means that they're overall more successful. Okay, and then going to some organisms that are close to my heart. I am a seagrass scientist for the most part, and seagrasses are a class of organisms where from basic physiology we might expect a totally different response. As plants, so seagrasses have kind of the same evolutionary trajectory as dolphins. They evolved out of the oceans and then evolved back into the oceans. These are plants that have pollen and seeds and lignified tissues. So they photosynthesize to make a living, and photosynthesis requires carbon dioxide. So acidification may be beneficial to photosynthetic organisms because it's providing more of this carbon dioxide that they need. And these same kind of laboratory experiments have shown that seagrasses do indeed either show no effect to acidification or do better under elevated CO2. And we have some really cool natural systems to kind of provide uncontrolled experiment about what happens in an acidified ocean. So there are a few places around the world, including some areas of Italy in the Mediterranean, the Ischia area, where there are natural seeps of carbon dioxide from the seafloor. So through geological processes, CO2 is constantly bubbling through the water, and this is a picture of a place where that's happening. And so here, independent of what we are doing to the oceans chemically, we have an example of a place that has super, super high CO2 concentrations, and we can look at what does that mean for the ecology of the area. And what we see here in an area where CO2 is bubbling through, the water is almost saturated with CO2, is that seagrasses and fleshy algae are dominant. Seagrasses do great. Algae that don't calcify do great. And calcifying species are absent from this area. So in Ischia in Italy, in general, sea urchins are very common and important members of the community, but they don't occur near these vents. And so that may suggest that they're sensitive to CO2. When you move away from these vents in the same area, you get a much more diverse community, and it includes animals like the sea urchins. Seagrasses are still there, but they're not dominant in the same way as they are around the vents. So this is consistent with the idea that seagrasses are going to benefit from an acidified ocean, and that what we might see is that there are winners and losers as acidification progresses, and overall there are going to be more losers than winners. So we're going to have less diverse systems as CO2 concentrations ramp up. So away from CO2 vents, you see more diversity, and seagrass does not dominate. All right, so now that's the background on ocean acidification, and trying to connect that to the Pacific Northwest and to the areas that DNR manages has been my task for the last year and a half. And the Northwest, as maybe a lot of you know, is kind of a hotspot for ocean acidification research worldwide, and in policy terms, actually. We are leading the nation and the world in the scientific response and the management response to this issue. So it's been a really cool place to work as this has become higher and higher in public consciousness. So the niche that I've tried to carve out for our research program at DNR has been to move away a little bit from these laboratory experiments where you have a tank, you pump up the CO2, and then throw some animals in and see if they die or not, and more into the field. So trying to connect ocean acidification science to the nearshore waters that DNR has a direct management interest in. And this is a complicated task because conditions are a lot more variable in the field than they are in the laboratory. And the other part has been trying to connect ocean acidification science, which has mostly been performed by oceanographers in the open ocean, to shallow bays and inlets of Puget Sound, which are very different environments. The main difference is that they're way, way more variable. So around Hawaii, where those data were generated that I showed you earlier, Hawaii is a really beautiful place and has a lot of diversity. And in addition to that, in some ways, it's a desert-like environment. It's very nutrient poor. That's why the water is so clear in Hawaii, and it has very low seasonality because we don't have snow melt out there. We don't have big seasonal rain inputs. So that means over the years, as you collect pH data, you get a pretty clean decline in pH, and you can say we've measured ocean acidification. In nearshore environments of Washington State, on a day-to-day basis, you see more variation in pH than the entire human forcing of the issue in the open ocean. So to date, we don't have strong statistical proof of acidification in nearshore environments. Some people would be mad at me to say that, but that's where we are, and trying to connect acidification to these highly variable environments is a tall order. They have. So one of the reasons why pH is so variable in the Pacific Northwest and in nearshore environments is because we're one of a few regions of the world that experiences an oceanographic phenomenon called upwelling, where really deep water in the Pacific comes up to the surface, and that deep water is very high in CO2 naturally. So for the past several million years, the Northwest has been an area where seasonally, usually spring and late summer, we have these pulses of very corrosive, high CO2 water that wash over our lands. And shellfish growers, indeed, a few years ago, had a few full failures in their hatcheries where they pulled water in from Puget Sound, or from Hood Canal, actually, that was where the hatchery was. And all of the shellfish larvae died. And they did a lot of experimental work to figure out what exactly about that water was wrong, and found out, this was a little earlier in the science of ocean acidification, that was the issue they hadn't been paying attention to, the buffering capacity of the water, the pH of the water. And they solved the problem in the hatchery setting in two ways. One of them is by changing the timing of when they pull in water. So being conscious of acidification as a problem for shellfish larvae, they now know that during these upwelling episodes, which happen naturally and which are fairly predictable, you don't pull water into your hatchery, because that's the time when the natural water will just kill your larvae. The other thing that they have done pretty successfully is artificially change the chemistry of that water. So instead of pulling it in directly from Hood Canal and dumping it on their larvae, they pull it into holding tanks where they up the buffering capacity by dumping limestone, essentially, large concentrations of sodium bicarbonate into the water, which makes it much more favorable for shell building. So that's part of the story for shellfish larvae in the hatchery setting. In nature, clearly we can't do anything like that. It's not realistic to dump sodium bicarbonate in like thousands of tons of it into a bay to try and protect shellfish larvae. So my focus has been since we can't do something like that, what can a management agency do about ocean acidification, which is a global problem. It's not something, even if Washington State decides to stop emitting carbon, that's going to be solved quickly. So I, having a lot of background in seagrass biology, started to look at this question of whether we can use photosynthetic organisms to locally soak up some of the carbon dioxide that we're putting into the water and improve chemical conditions at a bay-wide scale. So we know from laboratory experiments that seagrasses do pretty well with elevated CO2, and trying to move that into the field to see so we know how the environment affects seagrasses, but asking the converse question, how do seagrasses affect the environment, has been the first direction of my research. So to connect that to animals that are economically important, this is an aerial shot of Willapa Bay. Hopefully you can see all right. And oh yeah, here's the laser. So here's an area of unvegetated tide flat. This is an oyster farm. You can see clearly delineated along the lease lines. And this is a long line farm, so the oysters are held above the surface. And then over here is a seagrass meadow. So there are 3 different habitat types. And I wanted to ask, what do those different habitat types mean for the chemistry of water passing over a tide flat? So let's imagine that there's a geoduck larvae, which is microscopic. It's just carried by the current over these tide flats. What does it mean for that geoduck larvae to pass over this bare habitat? And by bare, I don't mean it's there are still things living there, but it just doesn't have oysters, doesn't have seagrass, versus that same larvae being carried over an oyster farm, or that larvae being carried by currents over eel grass. So, to approach this question, we just looked at basic physiology to come up with some predictions, and what we know about mollusks and seagrasses from first principles is that they both respire. So hopefully everybody knows this about plants, but plants photosynthesize and respire. To build their tissues, consume oxygen and produce CO2 just like we do. But plants photosynthesize and animals do not. And we know that on net, plants capture more CO2 than they emit. That's how they build themselves up. So, knowing this, that mollusks like us are respiring, putting CO2 into the environment, and seagrasses are soaking it up, our hypotheses are that local pH will increase as water passes over eelgrass and decrease as it passes over oysters with respect to the CO2 that's either coming into or out of the system. So we tried to pretend to be geoduck larvae. The way that we approached this question was by using a drifting approach. So this is one of our drifters, which we just let passively move through the environment. And I'm in a kayak right here taking this video. So I don't know if you can see very well, but this is the drifter passing over a seagrass habitat. And the way that we tried to analyze what was happening to water chemistry as it passed over different kinds of habitats was to set up our drifter upstream of a given habitat type. As soon as it reaches that habitat, we would grab water samples and preserve them to measure later in the lab. And then as soon as the drifter passed off of that habitat type, we would take another set of water samples, all the while having measured the position of the drifter and how long the drift took. And then by comparing the end versus the beginning water samples, we could see how does chemistry change as water passes in these shallow environments over these different habitat types. So it was a great project. Get paid to work on a kayak, move around Washington State. And the results of this project, which were 2 years ago, were very intriguing. So on average, having many, many drifts over 5 sites all around Washington State in a replicated experiment, we found that drifts, so this is looking, on the y-axis at the change in pH per hour of water that's passing over a given habitat type. So this is standardized to per hour because the drifts were all different lengths. And for drifts that happened over bare habitat, we saw no net change in pH per hour. Sometimes it went up a little bit, sometimes it went down. Here you can see the average is a little bit negative, but the error bars overlap with 0. So there was no net change in pH. For water that passed over seagrass habitat, we saw an increase in pH of about 0.06 units per hour. And that's a significant effect because so far, like I said earlier, the cumulative effect of human carbon emissions on ocean chemistry is about 0.1 pH units. So in 2 hours, seagrasses are counteracting that effect, or more than counteracting that effect. By pulling carbon dioxide out of the water, they're having a dramatic effect on local water chemistry. And we saw it in some of the laboratory measurements that we took after collecting these samples, that in really highly seagrass-dominated systems, like Padilla Bay, Samish Bay, Fidalgo Bay, in the middle of the day when the grass is really cranking, photosynthesizing in high light, you can see crazy pHs. The normal oceanic pH is between 7.9 and 8.1. In these eelgrass bays, you can see pHs of 8.8, 8.9, like really good shell building conditions. In fact, the grass in mid-afternoon stops photosynthesis because there's no more carbon dioxide in the water. It becomes carbon limited. There's plenty of light, there's plenty of nutrients, but there's no more CO2 for it to consume. So then what about our other hypothesis? We didn't find support for it. So there was also no net change in pH of water passing over oyster reefs. And after we found this result, we kind of looked back over past scientific work and found that maybe we should have done some back-of-the-envelope calculations earlier. But from what we know about how much oysters respire based on their biomass, we would probably not be able to detect change. That the amount of CO2 that they're putting into the water is vanishingly small, and we would have to have done probably 100 times more drifts to have the statistical power to measure that change. So they are putting CO2 into the water. it's not at a level that is going to do something at a bay-wide scale. So the conclusion here of this first phase of work is that eelgrass does increase pH through photosynthetic drawdown of CO2. So, looking back over here, we have our gooey duck larvae, we have 3 different habitat types. The conclusion is when a gooey duck larvae gets carried over eelgrass, it's being carried through an area of increasing pH that may shelter it from the effects of ocean acidification. So the next phase of our work has really been to elaborate on this finding. And one of the first things that we needed to answer was that we did all of these drifts in the middle of the day with lots of sunlight, with warm water, and like I said, plants just like us respire. And so at nighttime, there's no photosynthesis happening. The seagrass is actually putting CO2 into the water. So we need to be able to answer the question, maybe in daytime, seagrass is a great place for a gooey duck larvae to be, but at nighttime, it could be just as negative. It could be bad for them. So, implications here are that eelgrass could be a management option for mitigating ocean acidification. We can protect seagrass or restore it to places where it's been lost as a way to provide shelter for these microscopic larvae as they're experiencing elevated CO2. So this is where Cherry Point became a point of focus for us. So to answer this question of what is seagrass doing at night, is it counteracting all the benefit that it gives us during the day, we established a acidification nearshore monitoring network. Anemone is the acronym. So we have 8 sites around the state, and from north to south, these are them. And at every one of these sites, we have installed sensors and instrumentation to help us get at this question. Two different questions. One of them is, what is seagrass through respiration doing at night? Does it counteract the benefit that it has during the day? And the other is, what is the seasonal pattern of this seagrass benefit? In the summer, the plants are really active. In the winter, they're kind of in a quiescent state. They're hibernating, the plants. They don't photosynthesize very much. They're just waiting out the cold months. And so we want to know, when is the seagrass pH elevation significant? So at every one of these sites, we have sensors that are measuring pH, temperature, chlorophyll, dissolved oxygen, and salinity. And Eleanor here was helping me the other night swap some of these sensors out if it all go bad at 3 a.m. So this is what they look like. At every one of these sites, we picked them for a variety of reasons. We tried to have as much overlap as possible with the Aquatic Reserve system so that we can build on all of the great monitoring work that they're doing there. And we had to pick places that had both of these habitat types. Some areas with seagrass, some areas with no seagrass. And not every aquatic reserve meets those criteria in terms of the aerial coverage of seagrass that we require. So then at every one of the sites, we have these sensors installed with pH monitors, salinity monitors, dissolved oxygen, and chlorophyll. And we've deployed them now for a little bit more than a year and are building up this time series, which we hope becomes a kind of backbone for further scientific work. We're creating this network of environmental measurements where we would love it if people took biological observations that are co-located with these environmental parameters to help us understand what acidification means in these shallow environments where it's hardly been studied at all. Because even now, it's a slow process of bringing ocean acidification science into estuarine environments, into nearshore environments. Oceanographers still dominate this issue, and they're working in open ocean, really deep water. So to show you some of the initial results, this is a short time series, just a couple of days, and what we're going to look at is the pH anomaly. So this is in one site, and I pulled these data from Cherry Point to try and make it more relevant. At one site, what's the difference in pH between the area with seagrass and the area with no seagrass? And in this case, I did not expect there to be a huge difference because they're very close together at this site, and the seagrass is not dominant in the way that it is at Samish Bay or Padilla Bay. It's a patch of eelgrass that's not that big. And so we have our sensors in there, our sensors outside, and then we're comparing them. So here at the value of 0, that means there's no difference between seagrass and bare habitat. And over the course of these days, this is what we actually see. That these yellow areas, that's daylight. The areas without yellow is nighttime. And so during the day, even at this small scale, this eelgrass patch of only 20 meters in diameter, you have these really dramatic differences in pH. And you know, on a particular day, I don't know if this one was sunnier or what, but on a particular day at midday when photosynthesis is really going up, you can see pH values more than a unit higher than an area only 50 meters away that's outside of seagrass. And this is the beginning of an answer to our question that at night you do see lower pH in seagrass because it's respiring and putting out CO2. When you sum up these differences, there is a net benefit to being in seagrass. The pH elevation during the day is greater than the pH depletion that you see at night. So, eelgrass does appear to locally elevate pH in daylight and decrease it in the nighttime. So, this is the last part of data in the talk, and then I'll be really happy to take questions. But just last year, we've moved from these basic observations of the chemical effects of seagrass to try and connect it with the animals whose performance we're worried about, so bivalves, clams. And to get at this question, we took our observations to date, which is that our research shows that seagrass absorbs CO2 and increases pH, and try and answer this question of larvae, which are the most sensitive life stage of these animals. Once they settle down and start growing up, laboratory work has shown that adult and juvenile oysters and geoducks are way more robust to pH change than the larvae are. The larvae, as many of you probably know, have really limited energy stores. They're small. They have a short period where they have to make their way through the water and settle down to become adults. So it's crucial, the chemistry around them in this phase of life. And to answer this question of whether larvae exploit the positive pH of eelgrass, we reflected on, again, the experience of individual larvae. So earlier I kind of talked about a geoduck larvae as a passive microscopic thing that gets carried around by currents. And for the most part that's true, that these animals are so small that they can't fight against currents. They can't move upstream. What they can do is that they have the ability to control their vertical position in the water column pretty well. They can move up and down depending on a lot of different factors. And old laboratory work has shown a number of cues can cause these larvae to move up and down. Oxygen levels, temperature, salinity can cause them to move up and down, but they're not passive particles floating around. They actually have the ability to control themselves. And here is just a video of an Olympia oyster larvae moving around. So they are mobile creatures. They can't fight against the current, but if they detect something more favorable below them, they can pull themselves down towards that favorable chemical condition. All right, so we had 4 species that we picked mostly because they're economically important, where we wanted to answer this question. Pacific oysters, Olympia oysters, geoduck, and manila clams. And to get at this question, we have this hypothesis that larvae do exploit the better conditions in seagrass. And if this is the case, we're going to see different things during the daytime when photosynthesis is happening than we will at nighttime when there's no photosynthesis. And if we collect data to support this, then what we're going to see is that in daytime, larvae rush down into seagrass. They can detect that better pH inside the seagrass, and so they pull themselves down. In adjacent bare areas, there may be no pattern because there's no pH gradient throughout the water column. At nighttime, we might see an opposite pattern, that the larvae that are drifting around drift into seagrass. The seagrass is respiring and producing too much CO2, and they move up away from it. And again, at nighttime, there might be no difference in distribution vertically. So the way that we did this was, again, getting paid to go out in kayaks, going out both at daytime and nighttime with these portable battery-powered pumps on the back of our kayaks and collecting water samples at different depths. And then those samples were preserved, and we later looked at them to figure out, at each different depth, how many larvae do you see, to figure out whether there are indeed more of these species inside of seagrass in daylight. And to show you the very initial results, which are just coming out now from this project, these are for Pacific oysters only. And again, this is coded as nighttime, this is daytime, and I'm going to show you the proportion of the larvae in the different habitats. So this is kind of the first pass question. At nighttime, at a given spot, you see a lot more larvae in eelgrass than you do outside of eelgrass. And there are a couple of reasons why you might expect this, but some of them are just physics, that there's hydrodynamic changes inside of seagrass. There's a baffling effect that causes the water to move more slowly and small particles to settle down. In the daytime, you see the same pattern. So this was a little reassuring to us because it shows eelgrass is having this physical effect, but it's not, it doesn't change from daytime to nighttime. That in general, you find more larvae in seagrass. So then, from there, what can we learn about their distribution within the water column, higher or lower, with respect to eelgrass photosynthesis? So here in bear habitat at nighttime, this is the proportion of larvae, about 65%, that are low down, close to the bottom of the water column. And these are the proportion of larvae, 35% or so, that are up near the surface. In seagrass, it's not too different. So at nighttime, you find more larvae deep down, and there's no statistical difference in their distribution between these two habitats. Distribution did not vary across habitats by night. In the daytime, the larvae distribute themselves differently. They move towards the surface in bare areas. So you see a much higher proportion, actually basically an inverse proportion. 70% of them are up at the top of the water column and 30% low down. And in seagrass, we see a statistically different distribution, where in daytime, when there's photosynthesis happening, there's a lot more larvae down inside of the seagrass, which appears to support the idea that these larvae can detect pH gradients and move towards more favorable conditions. All right, daylight. Larvae in eelgrass were more likely to be deep. So that's where we are right now. We've moved on to some additional projects trying to come up with a gene expression assay to measure when these shellfish are experiencing stress from ocean acidification and climate change. And we're really eager to get these results on larval distribution for our other 3 target species. But with that, I think I'm happy to take questions if there are any. And thanks for coming out on a Saturday to hear about science. Yeah, I don't want to get too controversial. Definitely the direct suggests that there may be something to co-culture of shellfish and seagrass. And some of the work that we conducted this summer supports that idea, where we actually, for the first time, ran a large-scale experiment around the state and found that juvenile shellfish, Olympia oysters and Pacific oysters, grew significantly faster inside of seagrass, which we actually didn't even expect that. We didn't think there was going to be any difference. But there, more than just on the shellfish grower side, there are regulatory barriers for that happening. The Army Corps, for instance, now requires pretty substantial buffers between seagrass and shellfish farms. And for any sort of co-culture to happen, the regulatory regime would have to be changed. And then for the first part of your question, it's also an interesting issue, but there could actually be different cues that are followed by these animals with respect to their early larval stages. So early on, they're looking for good pH, they move into seagrass, but where they want to settle is actually based on something different. Like we know that Pacific oysters and Olympia oysters recruit to hard surfaces, which eelgrass, that's not where you find eelgrass. So it may be that early on in their life, these larvae are spending more time in seagrass to build up their shells and get ready for adulthood. And then they allow themselves to drift out, find a hard surface, settle down, and start making a living. Yeah. Yeah, it is. I'll repeat the question. So the first part of the question is whether there are differences in the effects on carbonate chemistry between the two seagrass species that we have here, Zostera marina, which is native, and Zostera japonica, which is not native. And all of our drift work was looking only at the native species, in part to not be controversial, to avoid certain politics. But the work that's been done to date shows that carbon assimilation rates for the two species on a per biomass basis are pretty much exactly the same. So we would probably see exactly the same thing if we drifted over Zostera japonica as we would marina. The only other small part of that question was, to me, the effects of these plants on carbon really do fall into two different buckets. One of them is the immediate effect of photosynthesis on the conditioning of overlying waters. So that's what we were trying to measure, the pumping up of pH on a short scale. The other is carbon sequestration, which is long-term storage of carbon in plant tissues. And that is also a really big research question that I haven't been involved in, but I would say that emerging evidence shows that seagrass is not great at that. That trees on land are pretty unique in the plant kingdom at their ability to build woody tissues that store carbon for very long periods. Seagrasses are super productive, they photosynthesize very quickly, but then their material decays almost as quickly as it gets grown. They don't build up huge trunks that are underground storing carbon. And then the second question was, variability in instrumentation. So that has been a big learning curve. For me, still currently, it's hard to buy commercial pH sensors. They're enormously expensive. So the way that we've approached the question is I've built all of our pH sensors, and we deploy them in duplicate in every case in order to show minimal drift between individual instruments and to document that the variability we're seeing is actually has high fidelity at the site scale. But we see huge variation. That's the issue of studying ocean acidification in nearshore environments. We're going to have to measure these conditions for many years before we can detect any kind of slight decadal change in pH. Because like I said, you saw it at the Cherry Point data actually. In a particular place, in the middle of the day, pH can be 1.5 units higher than it is at night. And that's an enormous range of variation. Yeah, so we realized after our first year that is one part that we couldn't really tease apart from our methods. So the fact that we saw no net change in pH in drifts over bear habitat means that in those bear drifts, there was phytoplankton in that water, algae that was photosynthesizing, but it clearly wasn't happening at a scale that had a difference that mattered for pH. To kelp, macroalgae, yeah. So we didn't drift over any macroalgae. There are a number of projects that are looking at the ability of macroalgae to have the same effect on pH. One of the issues there that's different with seagrass is that most kelp species are annual, so they're also tremendously productive, but then they die back entirely in the winter. Like Nereocystis, the bull kelp, grows up, forms these nice forests, and then it's completely gone. Whereas seagrass is there year-round. It's perennial and forms these stable beds. So they do have different implications for shellfish larvae in that respect. Yeah, we didn't. So there are a lot of things that vary at the same time as pH, and it's a really good point that these larvae moving into seagrass doesn't necessarily mean that they're freaked out by ocean acidification and rushing into great pH. They could be headed towards higher oxygen. Salinity is not likely because there's not any change in the salinity gradient between day and night. Oxygen and temperature are the really big co-varying factors with pH. And in a field experiment, you can't pull them apart. Yeah, but thank you for asking that. Mm-hmm. Yeah, it's kind of the same issue I think as the anchored kelp, macroalgae, that I think they really do have an effect, just because we know from lots of work that's been going for decades how productive these algae are. But they're going to be a lot more ephemeral in time and in space. The bull kelp go away during certain times of year. The sargassum aren't anchored anywhere, so they're having a big impact wherever they are, but then they're everywhere. So they're not the same, they don't have the same management promise that seagrass does, because you can take efforts to protect seagrass in one place, and then you know it'll stay there because it's a perennial plant, or you can restore it and then it'll be there. If you go to the lengths of restoring bull kelp, it's not likely to be there the next year because of its life history patterns. Yeah, there's two science teams in DNR Aquatics, and one of them is devoted to that question. And again, it's at such a big regional scale, it's hard to attribute it to any one thing, but there has been a slight increase in eel grass coverage in Puget Sound over the past 15 years. I would be pretty reluctant to say that that has anything to do with carbon dioxide. Oh, yes, sorry. So the question was about whether, with more CO2 in the water, that's beneficial to seagrass, whether we've seen seagrass do better over recent history. Yep. Yeah, so the point was that there are places, so sargassum does drift around, but there are also places, yeah, which I didn't mention, where sargassum is anchored and semi-permanent, and that those might be a similar place of high pH, high photosynthesis conditions, and that's worth looking into. The challenge, I think, across these different sites in evaluating the potential of aquatic vegetation, both seagrass and macroalgae, has a lot to do with the context of a given site, especially with bathymetry. So the places where I think there is really big potential to have positive conditions for shellfish larvae are shallow bays that have limited water movement, and where there's high entrainment, where the water comes into the bay and stays there for a long time. Cherry Point is a much more exposed place where water is coming through the strait and washing up onto a narrower fringe. And so you don't have the same proportional coverage of aquatic vegetation. So that means maybe right inside that sargassum bed there's really great pH, but it's probably not going to pump up the pH at Cherry Point broadly. It's where the Puget Sound Corps maintains their seagrass in that site. So it's in, yeah, okay, that's right. Yes, so the question is about devoting a little more attention to phytoplankton at Cherry Point specifically. So when we first put out these instruments at that anemone network of 8 sites, we did not have chlorophyll sensors. And the reason that we went to the trouble of writing grants to get those chlorophyll sensors is because we wanted to have at least a baseline level of knowledge about how much phytoplankton is in the water. So the chlorophyll is measuring indirectly the amount of phytoplankton and doesn't really have anything to do with seagrass. And one of the reasons to do that is because this first year and a half of data has shown that there really is a difference seasonally in what seagrass is doing. Over the winter, it's barely photosynthesizing, it barely has a pH benefit. And then in the early spring, March or so, the grass wakes up and starts photosynthesizing and has dramatic effects on water chemistry. And that is happening at the exact same time as the first phytoplankton blooms of the year. So it's, we need chlorophyll sensors to be able to disentangle that, to say, because we see this big pH increase, and it could be due to eelgrass, it could be due to phytoplankton, and only with a few years of data and sturdy chlorophyll measurements are we going to be able to break the pH increase into those two different actors, into phytoplankton and eelgrass. No, I'll be around later. I'm depending on other people for my ride. All right. Should we transition? More questions? Thank you.